And welcome back to the Behavioral Neuroscience lecture. In this section, we're going to begin covering Chapter 11, which is called Sensation, Perception, and Cognition. As you might imagine, the first thing we're going to start looking at is your sense receptors for the sensation section. And in fact, this material is going to overlap quite a bit with what you've seen in anatomy and physiology. So if that was material that you did well with, this is going to be a breeze for you. But there's not a whole lot of discussion of psychology at this point. We're mostly doing basic physiological descriptions of the sense receptors at this current time, and then we'll move on into more psychological process later. The first sensation to be covered is related to your somatosenses, or touch. Now, touch has a wide variety of subtypes. It's kind of funny how senses have generally been identified, that you'll say touch is a sense, but then there is detectors for pressure and vibration and temperature and pain. And if these occur on the outside of your body or the inside of your body, where they might be called organic senses, then they get classified as separate subsections of touch itself. Why not just call all of these separate senses instead of just making this general classification? That's a good question. I think it's just a historical aspect in which these things have been classified the way they have. One additional part, without you thinking that I'm just rambling on about historical aspects here and irrelevant things, is the fact that, as we're going to see later on, you will have a vestibular sense. And when we cover your vestibular sense, which is balance, that is formally your sixth sense. Okay? So if you ask somebody how many senses do you have, the proper answer to that is 6, because balance is your sixth sense. But the classification system of senses is really kind of historically willy-nilly. There's not a lot of specificity to it, so just accept it the way it is and don't question it. Somatosenses have a variety of subtypes. These are all fairly straightforward, so let's just take a look at them and accept the system that exists at the current time. Most of the somatosenses occur from the outer surface of the skin, and they do fall into categories of pressure, vibration, temperature, and pain. Those are fairly straightforward. Kinesthesia, however, is related to information about where your bodily movements are. So if you're in a completely dark room or if you're blindfolded, you can get an idea of where your limbs are in 3 dimensions by just feel. Kinesthesia is the sense of feeling where your limbs are, largely based on the stretching of your muscles and your tendons. Your organic senses come from experiences inside and around your internal organs, but those could involve pressure, vibration, temperature, and pain, depending upon what's actually inside of you. So the same receptors on the outer surface of your body are called cutaneous, but if they're on the inside and you feel pressure from your abdomen due to excessive gas, then it's called an organic sense. What's really different about it? Nothing. The location you're at. That's it. That's again more of the willy-nilly category system that has been developed for sensation. And nociception is related to free nerve endings and how they get stimulated during tissue damage. Nociception is, you're probably aware, related to pain in general. And these free nerve endings become activated at times during which tissue becomes damaged. So as they become activated, they specifically go to their own area of the brain that elicits a recognition of pain, and that's essentially why you can recognize some things as actually being painful because of the nociception receptors. For gustation, which is technically the detection of stimuli on your tongue, you go to different types of receptors themselves. These are essentially hair receptors, as you see in the diagram on the right-hand side. When you look at your tongue, you'll see these large disc-shaped spots, and people refer to those as your taste receptors. But the outer surface of that is actually not the taste receptor itself. You have to go to that disc, and then you will find a very thin cavern which food items mixed with saliva have to travel down into, and then hair cells from the gustatory receptors themselves stick out into that cavern and collect the substances that come down. Depending upon the shape of the molecule of the food item that was eaten, it may or may not stimulate a particular hair receptor, which then will generate an action potential and send a signal back to your brain for you to detect what you had actually eaten. So for anything that lands on there, including saliva and so forth, that will modify your experience of what you might call taste in general, but technically it's gustation. Smell works very similarly, as we're going to wind up saying. In fact, smell and taste are both called chemical senses because in both cases of smell, which we'll get to next, and taste here, you have to have a particular molecule that affects the hair receptor to generate the action potential. And because you're always dealing with the detection of chemicals, those are called the chemical senses. Now, the extension of this, olfaction, as I mentioned, works very similarly. But now those hair receptors are actually up in your nasal passage, far back where they will have a large area for the air to mix and flow so that the molecules in the air have more opportunity to land on these hair receptors. Typically, things that a human being can detect from olfaction have molecular weights of about 15 to 300. Outside that range, human beings just cannot smell things. And of course, this is going to restrict your experience of the kinds of things that exist because there are many different substances that exist on planet Earth that are well outside that range. But to be sure, your ability to smell something means that that something has actually gone up your nose. And that's an important aspect of recognizing that once you have detected something by smell, it's actually already gone into your body. And in some cases, if it is poison, then it may already be starting to produce damage. Now, the olfactory epithelium has about 6 million receptor cells, which is really quite a bit. And that is going to give you your opportunity for the degree to which you can detect things. Of course, things like dogs and bloodhounds that are trained to smell different types of odors have many more olfactory receptors. So your ability to smell as good as or to detect objects as well as a bloodhound is certainly going to be restricted by your own human biology in this particular case. But one of the things you do see in this case, which is a slight side issue, is that in the diagram here of where the olfactory epithelium is, as you go into the brain, your olfactory center is just on the other side of the cribriform plate, and you're not that far from your hippocampus, which is going to be, of course, as we've already mentioned, one of the areas that's closely related to memory function. And so the idea that smells work very well to elicit memories is true, and it's true largely because of the underlying biological representation of those areas. Smell comes in your nose, the olfactory bulb is just on the other side, and just next to that, you're going to wind up finding the hippocampus. Because these areas are near each other, smell can affect memory more intensely than your other senses. Hearing, or more formally audition, of course comes from your ear. Your ear has three general areas, the outer, middle, and inner. And there are three bones inside your ear called ossicles. You should be aware of the Latin terms malleus, incus, and stapes rather than hammer, anvil, and stirrup, which is oftentimes taught in middle school. So malleus, incus, and stapes, my inner sanctum, is a little mnemonic I've given you. This is a fairly straightforward story here, the outer portion of the ear. Where you probably are more likely to get a question on the MCAT is the anatomical structure of the cochlea. The cochlea is made from bone, and it cannot be unraveled as it's shown in the image here. But because it has that snail shape, just for purposes of clarification, you take the curled snail section of the cochlea and unravel it, which of course you can't really do. And what you wind up finding is that there's two hallways in the cochlea. One which information gets sent down, and the next one which it comes out. And it's the cochlea itself is filled with a viscous fluid that the sound waves travel through. And the receptors in your cochlea are actually in that center wall. And that's a very important point for you to grasp. I have to imagine that's going to be one of the questions you'll wind up getting asked. The sound that is picked up by your eardrum passes through the malleus, incus, and stapes and then presses on the oval window. The oval window is just generating sound waves through the cochlea, which travel down, if you follow the red arrow, all the way down to the end, and then turn the corner around the wall and then come back out where they will eventually reach the round window. The round window is there only so that vibrations in that viscous fluid can continue to generate more. Because if you mechanically eliminated the round window, it would subdue the amount of vibrations through that viscous fluid. Now, the wall in this very center of the cochlea is the location where your auditory receptors actually exist. And those receptors become active as a result of the flow of vibration through that viscous fluid. And then those neural receptors then wire themselves together, come out of the cochlea, which is not really shown here in this particular image, and then run off to the brain so that you get analyzed sound. Now, your vestibular system, which is directly related to the cochlea itself at the opposite side, is controlled by three loops. Each of the three loops, called semicircular canals, are in the three directions of three-dimensional space. So it's kind of an X, Y, and Z plane. And it's at the other end of the cochlea. And these semicircular canals are, of course, made from bone as well. Now, the operation of the semicircular canals for you to detect balance and movement operates essentially the same way as it does for hearing. These tubes are filled with a viscous fluid. There are receptors along the side, and then the jostling of your head essentially sloshes that fluid around and causes the receptors to generate action potentials, so that way you could detect forward or backward movement, rotational movement, and of course the direction of gravity. So if you were in a building that collapsed and you were disoriented, when you wake up, you're in a room, but it was completely dark and there was garbage all over the place and bricks and so forth, and you didn't know which way was up initially because you were confused, all you have to do is really kind of stay calm for a moment and pay attention to the direction of your limbs and the feeling from your head of which direction you feel your body's being pulled down into, and that will start to orient you toward the direction of up and down. And that's coming from this vestibular system in this case, which is, of course, detecting any kind of movement in the direction of gravity. And of course, gravity will literally still pull you toward Earth, and that's another way that you're going to help detect it. But that detection is going to be generated by your vestibular system. And vision. Now, vision is a fairly straightforward one. You've been seeing the eye since you were probably in grammar school. This is just an anatomical layout of the eye itself. There are a few things here that you should be aware of, as I still hear people make mistakes about this kind of thing. One, let's take a look at the conjunctiva. The conjunctiva is a tissue that covers the front of your eye and gets connected to the eyelids. So I do still hear sometimes people say things like they put in a contact lens and it rolled around to the back of their head. That could only happen if they didn't have a conjunctiva. And if they didn't have one, they would have tremendous eye infections all the time. So be aware that the outermost tissue for your eye is the conjunctiva. Underneath of that, it's the cornea, and then you have your iris. The iris is the colored section that makes a donut hole, and the hole itself is called the pupil. Underneath of that is the lens. The lens is connected to muscles. Those muscles will squeeze and contract the lens so that the light that passes through the lens will get focused as accurately as possible on the back of the eye at a specific area called the fovea, which is not really shown here very well, except there's a sort of an orange tannish circle in the back that represents the fovea itself. So the fovea is the place where light is always going to be focused. Doesn't matter where you look, light is always focused on that one spot. And of course, there's a spot where no light gets detected at all, and you can see that here because there is the optic nerve, and the optic nerve, as it comes together from all the cells of the retina in the back of the eye, as it leaves the back of the eye, leaves a section without any light receptors themselves, and that is going to be responsible for your blind spot. Now, there are a few terms here, like accommodation. Accommodation is simply the process by which the ciliary muscles will focus the lens. This is, when you talk about infants having bad vision on the day that they were born, there's nothing wrong with an infant's eyes on the day they were born. The problem is they don't have enough actual experience in controlling the muscles of the eye to focus the light on the retina properly. And that takes about 3 months for them to learn that. And of course, they're not doing it consciously either. It's that as the muscles operate, they will see either clearer or less clear. And then just naturally through the process of feedback, their muscles will begin to control the accommodation process properly so that the child can see most clearly. Now, on the retina itself, the back of the retina is like a radar dish. And the very center of that radar dish holds 6 million cones. And of course, cones are for color. That's the way to remember it. C for cone, C for color. You can see in red, green, and blue. Those are the primary colors that you can actually see. All your experience of other colors like yellow, there are no receptors for, but are just created by your brain through the function of separate receptors functioning together. Now, all of your cones for a color-seeing creature like ourselves only exists on the fovea itself. Outside of the fovea, you only have cells that detect brightness, and those are rods. So, in the simple description you hear, rods are for seeing black and white and cones are for seeing color. And that's essentially true. That's a little bit more simplified than you should know it now. Cones are really wavelength detectors and rods are amplitude detectors. So for any light is always a wave. Okay, light is always a wave, and a wave always has two properties, a wavelength and an amplitude. because light has two properties, light wave and light amplitude, you have two receptors, one for each of those properties. You have a cone that detects the wavelength, and you have a rod that detects the amplitude, which is going to be associated perceptually with brightness. So those cells in the back of your eye primarily are laid out so that about 6 million cones are in the fovea itself, the location where light is always focused. And then the remaining back of your eye has about 120 million rods. Why so many more, right? Why 20 times more? Well, because there's 20 times more space, right? You only have the fovea, and then they have all the rest of that radar dish to fill in, and that's just filled in with rods. So you may sometimes experience this or hear people talk about this with stars, and maybe if you have a telescope. If you're out on a dark night and you're looking at a star and you look directly at the star, it may disappear. And the reason is when you look at it, you're focusing the light on the fovea, which primarily has cones that only detects wavelength, not brightness. And because you're dealing with a light source that is very faint, there may not be sufficient brightness in that case going in your eye for you to see it when you look directly at it. However, if you look slightly away from it and focus the light of the star onto one of the rods, then you'll experience it. And it's one of those odd things that as soon as you see it, then you go back and look at it, and of course it disappears, but then you look away from it, and then it's there again. It's like it's stalking you. But it's the difference is really the types of cells in the back of your eye that are actually being used at the time during which you're observing the light source. Now, your vision is probably I would say the least well understood sense. A great deal about vision is known, but there's an enormous amount of complication because if you go to your retina, there's actually 3 layers of tissue that are detecting light. There's ganglion cells, the bipolar cells, and then there's the rods and cones in the back, the photoreceptor cell layer. And the photoreceptor cells only come in 2 forms, rods and cones, as we were just talking about. But then there's going to be, in terms of primates such as ourselves, 55 different types of cells inside of the bipolar cell layer, which is going to complicate the manner in which signals get sent back to the brain and what it actually does. A lot of this, though, is related to kind of the early evaluation of coloring, shading, movement, shape, and so forth. But that wiring in the retina is so small and so complicated, it's taking decades and decades for researchers to figure out exactly how everything's laid out. And then you get to the ganglion cell layer. Now, one of the things about the ganglion cells that you should be aware of is that whenever light affects a cone, the cone will be connected to one and only one ganglion cell. And that ganglion cell will go back to the brain and identify specifically where a point of light had occurred. Rods, on the other hand, are wired together. Multiple rods are wired together to a single ganglion. So some of the actual shape of objects, or to say this other way, the visual acuity that comes from rods in your peripheral vision is reduced relative to the light that lands on your fovea that hits a cone, because a cone is attached to a single ganglion cell, and that ganglion cell is going to tell your brain exactly where the light came from. But if light hits a rod, there's a grouping of rods that may have been hit, and so some of the visual acuity is actually lost as that group of rods sends its signals to one single ganglion cell. One of the other things about light that you see focused on in the manual here is the differences between nearsightedness and farsightedness. And that's a fairly straightforward thing. It's related to the degree of roundness of your eye and how well your muscles can go through the process of accommodation to naturally focus light on the back of your eye. And in this instance, a variety of lenses are shown, how they bend light into the lens so that an individual who's either nearsighted or farsighted can increase the chance that the light that is in the back of their eye is actually being focused onto the fovea properly. In an aspect that I think is a bit more important, and this may be my own bias, is recognizing the wiring diagram of the visual system. There's an aspect here that you need to be very aware of, and that is when you take a look at the image right-hand side of the screen, it shows that the left side of each eye is wired to the left hemisphere's occipital lobe, and the right side of each eye is wired to the right hemisphere's occipital lobe. So the occipital lobe itself is split across your left and right hemispheres as one large area. But the section on the left side of your eye is always wired to the left hemisphere, and the sections on the right side of your eye are wired to the right hemisphere. So if we play a horrible, disgusting game here, we go in with a nail gun and we shoot a couple of nail guns into the back of the person's head on the left side of the brain. What will happen is, once we damage that section of the brain, the person will lose half their vision. In fact, they'll lose the ability to see anything, and note my words here, in their right visual field. Because anything in their right visual field follows a pathway of light that lands on the left side of their eye. And the left side of the eye for both eyes gets wired to the left hemisphere's occipital lobe. Therefore, once we damage the left hemisphere's occipital lobe, the person loses the ability to see anything on the right-hand side. And the reverse would, of course, be true if we shot nails into the occipital lobe of the right hemisphere as well. Then the person would lose the ability to see things on the left-hand side. If this is not clear, pause the video, stare closely at that image, and trace through the red and blue to see where it actually winds up. But you will see things like this in individuals that have strokes. So, for example, whether it was a closed head injury or possibly a stroke, an individual could have damage to the occipital lobe on the left hemisphere, and what they wind up with is blindness on the right hemisphere. So, if after a stroke a person can't see anything on the right-hand side, you're largely guaranteed to recognize, even before you get to the expensive brain scan that you're going to have the person do, you know where the damage in the brain already is. It's in the left occipital lobe. And this is a part of what you refer to as neuropsychology, where you learn about the types of deficits that occur as a result of where the damage occurs in the brain. So the more you know about the wiring of the brain, the better you can predict the types of behavioral deficits. And in fact, neuropsychology has done quite a good job that, despite the fact that you do still need to follow up with a brain scan, especially because the technology is actually there, you could take all that knowledge of neuropsychology and get a reasonably good idea of where damage actually occurs in the brain by just evaluating behavioral deficits. And there's a wide range of things, not just vision, but also intellectual tasks, recognizing movement from things standing still, telling colors apart. There's all sorts of things. Anything that involves some kind of stimulus discrimination, recognition of information, learning, memory, all fit aspects of neuropsychology, so that with no advanced expensive equipment, you can actually get a reasonably good idea of where damage occurs in the brain and whether or not it's actually improving based on any kind of treatment that you may be applying. Now, perception. Perception is a term that comes after sensation all the time. Sensation always refers to the actual recognition of some sort of energy source outside your body that impinges on a receptor and generates an action potential. Sensation is usually seen to be kind of as a basic physiological process. Perception, on the other hand, is actually sensation plus any learning and memory that you already have. So you have to detect something, of course. You have to have light go in your eye to be able to see something. But having been an individual that has seen lots of stuff, seen this before and that before, and had experiences with them that induce memories of these events, it modifies your experience of at the current time of what's happening, and that's what's called perception. So perception is always sensation plus learning and memory. And in this short description of perception for vision in this particular case refers to depth perception. Depth perception comes about as a result of two basic physiological processes, retinal disparity and convergence. You've heard of retinal disparity, no doubt. The idea that you have two eyes and each eye sees a slightly different view of the world. Your visual system compares those two separate images and judges how far an object is from you. So, in fact, the easy way to remember this is if you have to rotate your eyes inward quite a bit, like you're going to look at your own nose, that's always something that's close to you. But if your eyes are rotated in such a way that they're focused essentially in the same pathway, so that they're kind of parallel, producing a parallel line from your pupil out into space, then the object is going to be further away. And that's what retinal disparity is all about. Convergence actually is an aspect that comes from the muscular activity of your eye. And for some reason, you just don't hear about convergence much. But what convergence is literally the fact that you have to rotate your eyes all the time to be able to see objects close to you or further away. And with no conscious input on your part, and without anybody ever teaching you this from the time that you were an infant, the muscular activity of your eye gets eventually picked up by your occipital lobe as an additional cue to determine where things are, close or far away. And so if you were to take a muscle relaxant and to stick it on a person's eye so that they could not rotate their eyes very well, they would actually lose some of their depth perception because they would not have that additional stimulus being sent back to the occipital lobe, helping determine whether or not an object was close or far away. Now, if you've got one eye and you only have got monocular cues, can you not see distance? Well, absolutely you can, because there's many different types of things that you can use that are not binocular or retinal disparity or convergence, but that can come from a single eye. So, for example, relative size. Objects that are further away throw their light into your eye at a different angle than objects that are closer to you. In fact, things that are closer to you always appear larger. Things that are further away, of course, always seem smaller. And as a result, that's one basic way that you can use a monocular cue to determine distance. Interposition. Interposition is when one thing that's closer to you blocks your image of something else. So typical, like for a teacher in the front of the room, if a teacher stands behind a desk, the desk blocks part of the view of the teacher. That's a straightforward thing. You wouldn't even think about it, so obvious. But that also indicates distance. What's closer to you, the table or the teacher? Well, the table is because part of the teacher disappears because of the presence of the table itself. Relative clarity is also very similar to relative size. Objects that are closer to you always have more detail, and things that are further away always have less detail. And therefore, any object that you can see more detail of is more likely, based upon normal functioning of light, closer to you than an object that has less clarity. Texture gradient is exactly the same thing as relative clarity. I'm not even sure why psychologists make a distinction between these, but they are referring to the texture that you can see in an object. So for example, if you look at your t-shirt, for example, that you're probably wearing, you can see a lot of the detail in the fabric itself. And if you've got really good eyes, you can see some of the weaving of the t-shirt itself. But if you took that t-shirt and you moved it across the room and looked at it, it's just a blue t-shirt or a red t-shirt. You can't see any of the detail in it. That's the same thing as relative clarity, but for some odd reason, psychologists have made it a separate category to talk about texture, but it's nothing more than relative clarity. So be aware that those are just two categories that essentially overlap quite a bit, whereas relative clarity just refers in general to the amount of clarity you see. Texture gradient specifically is specifying the amount of clarity in the underlying texture of the object itself. Those are, to me, those seem to be the same thing. Relative height as well. Objects that are closer to you are larger than objects further away. And that goes back to relative size, but in this case, you are dealing specifically with height in this case. So if you were looking at telephone poles down a long road, the telephone poles that are closest to you are going to appear to be largest in your visual field. Same thing as relative size. Again, it's odd to me that psychologists make distinctions between these things, but it is what it is, and learn it, get yourself in a medical school, and be done with it. Relative motion. This one certainly recognize. If you're driving down Interstate 95 and you look out the window straight down at the road, it's of course going by at 70 miles an hour, which is of course really, really fast. Then you'll look up at the moon, and it looks like the moon is staying still. In fact, the road just underneath your car appears to be going 70 miles an hour as you're going 70. But then the side of Interstate 95 doesn't appear, especially on the opposite side of 95, doesn't appear to be moving as slowly. The houses and buildings just off of the highway appear to be moving slower still. Things further off in the distance appear to be moving even slower. And of course, the moon or the sun appear to just be standing still. Anything that's closer to you appears to be moving more rapidly. And the reason why this is occurring is because of the overall size of the context you're looking at. When you look out of your car right at the road while you're driving 70 miles an hour, you only see a few feet wide, maybe 6 feet of vision hitting the ground, and something that goes by at 70 miles an hour that only has 6 feet to pass by goes by very quickly. But then you look at the buildings way off in the distance. You're not looking at 6 feet any longer. You're looking at possibly 10-mile stretch in your visual field. And so a tiny section of 6 feet that appears to go by really fast on the road is not what you're really perceiving when you look further away, because you have a much larger context of everything you're seeing. And so the amount that you're passing by at 70 miles an hour that's 2 miles from you appears to be moving much slower, because its context within the overall visual field that you have is actually much smaller. Now, linear perspective is one where usually the easiest way to see this is with train tracks or even a road that you could kind of even see down here in Florida, like State Road 7. It's very long and straight. Of course, the road is 6 lanes wide where you're standing, so it seems wide, but when you look way off in the distance, it kind of comes to a point. If you look at a long sidewalk or train tracks, everything that gets further from you always is smaller. That was a story with relative size that we already talked about, and even relative height. But now we're applying it to the lines of the object down the road itself. So are you really looking at something different here? Not really. Again, it's kind of an overlapping concept with these other topics. Linear perspective is just telling you that the distance of the train tracks that you're standing at appear to be larger where you're standing, and because as things get smaller as they move away from you, those two lines appear to be coming together to a particular point. But that's because things that are further away from you appear smaller simply based upon the classical physics of the direction that light bounces around and eventually goes into your eye. It must come in at a sharper angle if it's further away to get into that tiny little pupil of yours. But when you're standing on the train tracks and you're looking straight down, then there is a wider angle that the light is coming from the train tracks that are on your left and right hand side. And of course, light and shadow. Light bounces around equally, essentially everywhere. Light follows the same properties everywhere it is. And for you to see something, light has to go in your eye. Now, statistically speaking, if you're in a gigantic room where it's equally lit every single place in the entire room, so it's like your physics experiment, the giant room as big as a gymnasium, for example, is equally lit everywhere. It is statistically more likely that light near you will go into your eye, and light from further away will less likely go into your eye. As a result, your perception of the brightness of that room, which you know is equally lit everywhere, will always appear to be duller on the other side of the room and brighter where you actually are. And that's, again, simply because of the amount of light that goes into your eye. Statistically speaking, light that's near you will have more chance to go in your eye than light that's further away, as it's all following the same principles bouncing around. So anything that gets further away from you always appears darker and darker. And that's just a normal perceptual experience based completely upon the straightforward functioning of light traveling in angles, and there only being a finite number of particles of light that are bouncing around inside the room. And another aspect of perception is certainly related to absolute and difference thresholds from the whole concept of signal detection theory. Signal detection theory is a process developed in the 1950s, which is really the formal way in which sensory and perceptual experiences are evaluated in a laboratory. It's a very hard-nosed, clear-cut experimental process. It gives you very good information regarding what a person can perceive, what they can actually sense, the activity of the receptors. It comes along with lots of mathematics. So it's actually a very well-developed field of study. In this particular case, what you need to really grasp is the differences between the absolute and difference thresholds. Absolute thresholds refer to your ability to distinguish the least amount of energy of some sort. So let's just say, for example, vision. What is the least amount of light you can see as a human being? So if we put you in a dark room, closed it off. You were in complete darkness for 20 minutes, so your eyes became dark adapted. And then we started to turn on lights of different sizes and colors and brightnesses and varied all the number of factors that related to light to see what was the least amount you could see. That is what an absolute threshold is. Now that, of course, is, it's interesting and it helps you understand the sensory system, but it doesn't play out very well in a practical sense because you're almost never in a scenario like that where there's absolutely no light and then you might detect some. You're more likely in a scenario of a difference threshold, which is the exact same story I just told you, except there's already some light in the environment. So if we stuck you in an enclosed chamber that was cut off from all external sources of light, but we stuck you in there with a 25 watts of light, we'll say, a dim light bulb. The fact that there's already light in there is going to affect your ability to notice any increases or decreases in light that occurs given the fact that light is already there. And that's the issue with the difference threshold. To go to extreme cases, if it's July, August, September in South Florida on a sunny day, and you're trying to tell the difference in variations of a brightness of your cell phone when you're looking at it under the bright sunshine in the middle of the day, you're going to have a much more difficult time than if you go inside of a building where there's not that much light and then evaluate the brightness of your cell phone, right? And it's easier to do it in a darker environment than in that super bright sunshine. And that's exactly telling you just what the story of difference thresholds are. The brighter it is outside, the larger the changes in brightness on your phone actually have to be. But if you're in a duller environment with less light, so you go inside of a building, then you can detect smaller changes on the brightness of your phone itself. The last point here is about Gestalt psychology. And Gestalt psychology is actually a rather big topic, although it's dealt with in a very straightforward, simplistic way here. When psychology got started, it was started in Germany. And so all of the initial terms in psychology were originally German. Anytime you see a German term in the field of psychology, it's almost a guarantee that you're talking about something from the late 1800s. The term Gestalt was a term that referred to recognizing the whole, the everything of a stimulus. And what it really is saying is that every stimulus occurs within a context, and the context modifies your ability to understand the stimulus itself. Consequently, Gestalt psychology was all about perception and how you cannot perceive something by itself. You can only perceive it within its context. And that's the primary issue. The image on the right-hand side of the page shows a Dalmatian in this odd image that's made in high contrast with the black and white only. You may have a little difficulty seeing the Dalmatian, the dog, but if you don't see it, I'll tell you where it is in a moment. Just give you a chance to turn the video off if you haven't seen it and you want to look for it. The dog is facing away from you, and its head is in the very center of the screen. Its head is down like it's sniffing the ground, and its body and legs are kind of coming back toward you, toward the right at about a 45-degree angle from where you're looking. And you probably pick up the dog if you take a look at it a little bit closer. But that largely covers this particular section. We'll be moving on to the next section on attention in the very next lecture.